brain, maybe something else. Yeah, brain, maybe macrophages. Yeah, okay. So microglia, they are brain cells. So they are the resident macrophages of the brain. So they are found, of course, in the central nervous system, so the brain and the spinal cord. Please tell me if I talk too fast. Just interrupt me, or if you have questions, please ask. Because normally I have the intention to speak quite fast, so please tell me if it's the case. So in the brain, you find a lot of neurons, of course, and then the glia. More than 50% of the brain is constituted of glia. And in the subdivision of glia, we can distinguish the macroglia, which they include astrocytes, oligodendrocytes, as you can see in this picture. So the neurons, the astrocytes, also ependymal cells. So these cells, they line the ventricles, and they also produce the cerebrospinal fluid. Of course, there are also these perivascular macrophages and endothelial cells. So then, apart from the microglia, you have the microglia, and they substitute, so they contain almost 5 to 50% of the total amount of glia, which is quite a lot. So, as you have all heard before, microglia are the innate immune cells of the brain. So that was the general view of microglia before 2000. So they are the immunocompetent cells. So they have a role or function in the protection of neurons against pathogens and damage, and they have their role in neurological diseases like Alzheimer's and MS. This is quite abundantly documented. So how now do microglia can protect these neurons, can protect the central nervous system from damage? So a lot of years ago, scientists thought that microglia were just sedentary cells, so they were just sitting there in their place doing nothing. So as you can see, microglia have very small cell somas, and they are highly branched. But until later, there was a study which did live imaging, so time-lapse imaging. So they imaged the microglia, and the study found out that the branches of the microglia were constantly scanning for insults, constantly scanning for damage. So actually, these cells were first called resting cells, resting microglia. But actually, this term is not effective anymore because they constantly scan and they have highly dynamic branches. So we will further refer to these cells as serving cells. So this model of microglia, so the ramified morphology, so which is normal in the healthy brain, which you encounter in a healthy brain, is not that, so how should I say it, the activation of these cells is not, this was just to indicate the scanning field that they can adopt. So actually microglia, they scan the whole brain in just a few hours because they are so dynamic. So if you look further to the classical model of microglia activation, so as I told you, they are present in the central nervous system in this resting, serving state, so highly branched. Then when there is an activating stimuli, the microglial cells will be activated, so they will retract processes, so their protrusions. They will become more rounded, so they will achieve more amoeboid morphology. And then they will start producing cytokines, and this will eventually lead to danger. So this was just a classical model of microglia activation. But this is no longer appropriate because there's also a lot of ongoing debate. Microglia, are they now beneficial or harmful in disease models? So a lot of contradictory results were found. So this model is not longer appropriate. Actually, microglia activation is quite dynamic and diverse. so than first anticipated. As you can see this figure from the publication of Hannes and Edelman, these persons are really the masters of the microglia. They've done a lot of research about them. So microglia are initially in the surveillance state. So they are resting, just scanning for possible damage. So now when microglia lose calming signals, or when they get activated signals, they will transform into an alerted phenotype. But from this point on, microglia can go out several ways, or they can adopt several phenotypes, depending on the stimuli, the intensity of the stimuli, the environment is such a big diversity. So microglia can adopt several phenotypes, then afterwards they can be reprogrammed, they can just die or integrate to another phase, or keep some memory function, or just differentiate back to the resting state. So now I would like to pose a question. Do events in living creatures happen without reason? What do you think? So are they just there with no function doing nothing? Of course, for bad events, this is obvious, because bad events, they will be filtered out through evolution. But even if microglia should be there and they are useless, even useless events or cells, they will also be filtered out because they cost energy. So these cells must have a function. So now in healthy adults, we know microglia, immune cells, infection. So they serve to regulate or modulate infection responses. But now, what is the case if there is no infection in the central nervous system? Why do we have these cells? So this is an example of a reaction of microglia to damage in the brain. So brain damage, it's not the same as infection. So if it will work, you should see a movie of microglia. You see the processes are quite dynamic, and you see this is a laser-induced lesion. And you see that the microglia, so their cell soma, stays stationary, and they just send their processes to the site of the lesion. So to form a barrier between the site of damage and the healthy neurons. So of course, now you might think, okay, laser lesion is not quite representative. This just not happens in normal people. But actually, micro damage can occur quite frequently, because micro damage may mean that it may result from tiny lesions in the blood-brain barrier, or little openings in the blood-brain barrier, and then leaving all these compounds from the blood coming in. So microglia can actually react to that. Or from small ischemic results. So micro damage is actually frequently present, and no one might notice. So microglia, they serve actually to constantly repair these minor insults. So now, of course, I told you something about the relevance of microglia in adults. But now microglia are also present in the embryo. So why is it? Is it just coincidence? So, because why are they present? So why are they actually needed? Because at that stage, the microglia or the embryo is protected by the immune system of the mother. So why should we need that microglia in the embryo? So therefore, the immunological function is less obvious. So we should search for other functions. So they have to be present there for a reason, otherwise they wouldn't be. So now I will focus on this. So from this part on to microglial functional roles in physiology in the embryo, and there are also some parts present from the adult. So roles during in adult organisms, and this is interesting because the functions of microglia in embryogenesis could also be interesting targets for therapy and education. So the ontogeny of microglia, which means the origin and development of these cells, it is highly conserved among organisms. So from the chicken to zebrafish to rodents and humans, it is all well conserved. So recently, it was established that microglia derive from the yolk sac. So from primitive, you will have these slides, a few slides further. So derive from primitive myeloid progenitors. So then they colonize first the brain, and then of course they are called microglia. They colonize the brain even before the liver and the lungs. If we now take a step back to embryology, maybe you remember. So the three germ layers from which an organism develops. So in blue you have the neuroectoderm. So neuroectoderm gives rise to skin epithelial cells and to neuroepithelial cells, which eventually gives rise to almost all cells of the central nervous system. Then in red you can see the mesoderm. Mesoderm gives rise to bone, muscles, cartilage, and the vascular system. And then in yellow, the endoderm gives rise to cells of the respiratory tract and cells of the gut. So here you can see the embryonic development. Here you can clearly see the yolk sac. So where these microglia are born as primitive progenitors, myeloid progenitors. So you see the yolk sac consists of a layer of mesoderm and an inner layer of endoderm. So microglia are thought then to arise from this mesoderm layer and then migrate to the brain and then penetrate the brain or enter the brain. So this is an image of a mouse embryo. So E, the letter E, means embryonic day. So a mouse, the gestation of the mouse is approximately 19 days. So at around day 8 of embryonic development, the embryo looks like this. And in blue, you can see a LAC-C expression of these primitive myeloid progenitors. So at this age, these primary macrophages are only present in the yolk sac. If we look one day further, you can see, here is the yolk sac open, that they already are present in the brain unit. So how can you now distinguish microglia from macrophages? I don't know if you maybe already heard about this, maybe from Niels Hellings or Janine Hendricks. So of course you can look at the tissue localization. You know, if you look into a healthy brain, that only microglia are there and no macrophages. You can also distinguish them via CD45 expression. So if you do flow cytometric analysis, you will find microglia or CD45 low or intermediate, and the macrophages will be CD45 high. So that's a way of distinguishing them. So this is just a slide to illustrate to you the embryonic development of an embryo. So it uses the Tyler staging. You don't need to know this, it's just an illustration so that you have an idea about the development of the mouse embryo. So now this part is important. I will show you some techniques how to visualize microglia, how to study them. I will mainly focus on fluorescence because that's the most important and widely used technique to image the cells. So I will first discuss some schemes and then I will briefly recapitulate everything the advantages and important things or the ways you can image microglia. So for example, the most widely used model of imaging microglia is the transgenic mouse model. So it's a mouse model in which all microglia, macrophages, T cells and subsets and dendritic cells will be green fluorescent. So the enhanced green fluorescent protein is expressed under the fractalkine receptor promoter, so which is CX3CR1. So if you cross then a homogeneous mice with a wild type one, you just generate heterozygous embryos which all have fluorescent microglia. So it's quite easy to study them, of course. What you can do then now, so this is the ex vivo setting. I included this slide as an extra one on Blackboard because there was a problem with the PDF printing. So you should have this one as the extra slide normally. Okay. So this is the ex vivo situation. So you cross these mice, you generate these embryos, you take them out. That's an ex vivo experiment. What you can do then is, on one hand, you can fix the embryos, fix the embryonic heads. So use paraformaldehyde, so you cross-link everything. So you actually kill the cells or cross-link everything that it stays in that position. What you can do then is make sections, thin sections of the brain, and then apply a lot of antibodies or lectins to label microglia. So antibodies, for example, against EBA1 is a general microglial marker. There are a lot of other markers also for activation levels. And lectins, a lectin is actually not an antibody, but it's a lectin which recognizes sugar residues on the microglial surface. So it will also label microglia. And then with just normal wide field fluorescence microscopy, you can study these sections. On the other hand, what you can do is, if you have isolated the embryo, you take out the brain by dissection. So you keep it alive, that's very important. And then you make acute brain slices. It's important that these slices are quite big. So these are 300 micrometer in thickness. So what you do is you embed the brain in an agarose matrix. You cut thick slices, you keep them alive with medium and oxygen and CO2. And then you can use time-lapse imaging so to actually record the behavior of the cells. So this is done by 2-photon excitation. So you should, I think, have an idea what it is or why it is used from the previous years. So, but briefly, 2-photon excitation, we use it because it is has a very low phototoxicity. So there are less radicals generated. You have very good penetration depth in the tissue. So you can image thick tissue, so image deep into the tissue, because it uses infrared wavelength, so wavelengths shifted to the infrared, and it has a very low scatter. So this is actually the gold standard for doing time-lapse studies in thick tissue. Then another possibility is the in vivo or the in vitro way. Of course, then you also can make use of this transgenic mouse model, but this is quite a complex experiment because you need to anesthetize the mother mouse, keep it alive. You have to take out the uterine horn, make sure that the embryos stay in the right place. So you embed the embryos also in an agarose shell, but it's a quite complex experiment, and then you can just in vivo image the microglia without cutting anything. So this is the best way actually to study the most physiological behavior of the cells. So briefly recapitulated, so ex vivo, what can you do? Immunohistochemistry, or for example also electron microscopy is also a way, but then you don't use fluorescence, of course. So immunohistochemistry, you have to know that it is static. So everything is dead, is killed. You won't see anything moving. You use thin slices for this because then you can easily use antibodies. They will easily penetrate because the slice is quite thin. So, and then, of course, you can make use of the transgenic mice to visualize microglia. So you can have transgenic mice and use a lot of antibodies specific for microglia to stain them. So the other way is the time-lapse imaging. This is the dynamic technique. It's functional because you actually see what's happening. You see the cells moving. It isn't functional of time because, of course, because it's a time series. You have to make acute brain slices for the ex vivo time-lapse imaging, and for that reason, it's more easy to work with transgenic mice because in these thick slices, antibodies it is very difficult for them to penetrate. So that is also why the use of transgenic mice is the best, and also the use of isolactin is also okay for this purpose. It will stain the microglia and also endothelial cells. What is a limit or a disadvantage about this ex vivo time-lapse imaging? Because you have to make slices, you actually damage the tissue. So when you make slices, you cut neurons, you actually let die some cells. So microglia, because they are so sensitive, they will activate at the cutting surface of the slice. So what you will do is then image more deep into the slice, and then it's okay. Another way of visualizing microglia, if you don't have a transgenic mouse model or if you don't have isolactin before, you can also electroporate a slice with a specific DNA encoding for fluorescent protein under the promoter of a microglia specific gene. So then also you can make the cells visible. Then the in vivo or the in utero setting, of course, it are all the same advantages as I told you before. But in this case, you have the whole embryo in its physiological environment, and you also don't have the activated microglia because you don't cut anything. But a disadvantage is that it's a complex experiment, and it takes a lot of time to set this up. Of course, also in utero, if you want to electroporate, then you have to do this in utero. So you have to operate a mother mouse, do the in utero electroporation, put the embryos back, wait a little bit. So you can imagine it's a very complex experiment. A lot of embryos die. So now, when we have a look back at the embryology, so this is just a scheme to illustrate to you how the cortex of rodents looks like at day 10 of the embryonic development and so on. So you see at E10 the cortex is quite thin, and it gets thicker, indicated by the C, when the embryo ages. So this is an example how what happens with microglia upon aging or upon maturation. So microglia, they arrive, as I told you, so they originate from the mesenchyme, they arrive in the central nervous system, and they are thought to enter the central nervous system via the pia, and also via the ventricle, and in a later phase, possibly via blood vessels. So they arrive there in the amoeboid form, so in the rounded form, without all the branches, without all the protrusions. So from this point on, they can migrate in all directions, so just to eventually colonize the whole central nervous system and take in their eventual mix. So as you see, microglial cells will differentiate over time into these highly branched, ramified microglia, as I told you before. So of course, in the beginning of development of the central nervous system, these cells are highly mobile or motile because they have to migrate to the places, to their eventual places. So when the embryo matures, the mobility of the microglial cells increases and the ramification improves. So this is what I'm showing you now is a time-lapse movie I made myself. So the question was here in the article that can microglia enter the central nervous system via the pia? So we made a time-lapse movie. So just for your orientation, so this is the image area, so the cortex with the pia, which you can see here, just on top. So I hope it will work. So you can see here the pia, the cortical plate, and then the intermediate zone and the ventricular zone. So you see in this movie that microglia just come from the pia into the cortical plate, or migrate just back from the tissue into the pia. I know that the quality is not that good anymore because of the projection, but you see that the cells, this one just migrated into the pia, other cells migrate out of the pia, so this is actually true. We saw that it happens. So now you might think, these cells, are they activated because they still have this weak morphology? But as I told you before, it's a normal process that the microglia differentiate so that they achieve the ramified morphology. And if you stay for activation markers, for example with MHC2, they are just not activated. It's just an embryonic phenotype. So now just to show you about the branching, that the branching increases. If we take a time-lapse movie one day later, it's maybe not that clear, but you can see that some cells are a bit more branched than others, so that the branching already increases. So now the pia is located somewhere here because the cortex is a lot thicker. What is important if you want to study these microglial or this behavior of microglia? So because almost nothing is known about how these cells behave, and you can deduct a lot, or can deduct something about their behavior. So what's important to study microglial behavior is to look at motility. So motility or mobility. So how they migrate, how they behave. And some things you can analyze are so the migration of the microglial cells, so the movement of the cell soma, and also the dynamics of the processes, so their protrusions. So how fast do they send out protrusions? How fast will they retract? How many protrusions do they send out? What angles do the protrusions make? The speed of migration, so the speed of cell soma migration, the speed of retraction, and so on. So now we've come to the second part of the lecture, which is about the other functions of microglia and during development. So microglia are non-randomly distributed in the central nervous system. So, which suggests that at these points or at these places they have specific roles. So microglia, for example, they concentrate in areas of cell death. So what could this mean? So yeah, they associate with neurons undergoing developmental cell death in the hippocampus, in the cerebellum, etc. What could this mean? So because they are closely associated, it could mean that they phagocytose developmental, that they phagocytose these apoptotic cells. And I can show you, I hope it works, a movie of, so in vivo imaging of a zebrafish embryo, normally in which you can see a microglia phagocytosing neurons. So this was the video. So in green is a microglia, in red are the neurons, and this is yellow because of this co-localization. So you can see here microglia forming a cup-like structure and then pulling towards it the neuron which is inside. And the other movie is about the digestion of the neuron inside the microglia. So here you can see these amoeboid microglia. I know the movie is quite short. You see that the red neurons are present inside the cell that has just digested the material. So this is really in vivo evidence of microglia phagocytosing developmental apoptotic neurons. But of course, what's also possible is that they just are not implicated only in phagocytosing these apoptotic cells, but that they also induce apoptosis themselves, so that they can kill the neurons themselves. And this is also true if they can instruct cell death via TNF, NGF, and respiratory bursts. So now, microglia are not only present in regions with apoptotic cells, also in regions where there are no apoptotic cells. So they also have function besides instructing cell death or apoptosis and phagocytosis. So they are also found in proximity of blood vessels. What could this mean? I told you before that microglia, they probably also enter the central nervous system via blood vessels. So there are some scientists who say no. So there are some scientists who say yes, microglia enter the central nervous system via blood vessels, because if we make an embryo without a heartbeat or without a functional circulation, there are no microglia present in the brain. But there are also other scientists who say that microglia are already present in the embryo before vascularization even happens. So probably it's a combination of these two mechanisms. So for the cortex of the mouse embryo, microglial colonization precedes this vascularization, so they are already present there before blood vessels develop. And in a later phase, they could enter the central nervous system via blood vessels in the choroid plexus. So that's one part. So they are located close to blood vessels because they enter via blood vessels, or they may enter via blood vessels. Another way of thinking is that they have a role in angiogenesis. So microglia are closely associated with blood vessels, and they also have a role in angiogenesis during tumor development and wound healing. So that's why hypothesis is in favor of that microglia promote angiogenesis. So evidence for this is that if you scientists who knocked out microglia, or knocked out actually the transcription factor PU1. So PU1 is the transcription factor. It's a key regulator of the myeloid differentiation. So embryos or organisms without this transcription factor do not have macrophages or microglia. So they see that the vascular network is very more thin and less dense than in normal organisms. So that is why they think microglia have an important role in angiogenesis or the development of blood vessels. Another point of evidence is that microglia facilitate tip cell fusion. So tip cell fusion is important when blood vessels develop, so they make anastomoses. So these cells, which you can see here, they are called the stalk cells. And then these tiny stripes here, these are called the tip cells who will make the anastomosis. So microglia, which you can see here in green, are located at the most important sites to mediate or to make the connection between the tip cells. So this is a 3D volume rendering image of this one. So this is actually a Z-stack made. So if you all know about morphocal microscopy, you can make these optical sections. And when you record all these optical sections, you can make a 3D image of it. So you can see here that microglia is around the tip cells, connecting them. Another point of evidence is the aortic ring explant. So what did they do in this case? They took a piece of the aorta from neonatal mice. So they took out a piece of cross-section, they brought it in a culture together or without microglia. You can see here that without microglia there is far more or less sprouting and branching of new blood vessels than in the case with microglia. So now, almost finished. Microglia also concentrates in a cellular space with developing axon vesicles. So we will now look more into the role of, or into the interaction between microglia and neurons. Actually, I have to confess that there is not a lot known about this topic. There is a lot known in vitro, but you have to be very careful taking in vitro results for reality. So we know that microglial conditioned medium enhances neuron survival. It also increases neurite growth and complexity, and it promotes neuronal maturation. So this is what we know in vitro. And the only thing we know in vivo is that microglia just concentrate at sites of dendritic growth, and that they associate with optic tracts and axon bundles. So that's practically everything we know for sure. So in the adult brain, of course, it is known that microglia can sense neuronal activity. So in this picture, you see that at a certain time point, TTX, which is tetrodotoxina, will, you've heard about it before, normally in your first year. It's a blocker of sodium channels. So if you give this TTX to the neurons, they will stop firing. So you can see here, for yourself at the time point, around now, it is added, and you see that the microglial processes just start to retract, that they are not that dense anymore because they don't sense neuronal activity anymore. And this table, of course, you don't need to know it by heart, it's just to illustrate that all these compounds microglia are sensitive to. They can react or they can sense all these compounds. So it's very likely that microglia really can sense neuronal activity. So now a little bit more about the neurics development. So I think so this is postnatally. A study has seen that microglia engulf non-myelinated fibers at a period when some transitory axons need to be eliminated. And this is studied in the adult. So in the adult there is a permanent contact of microglia with synapses, and the contact depends on the visual input they get. What is more, microglia postnatally also actively phagocytose synapses. So this study detected pre- and post-synaptic material inside microglia, so which is evidence that they really phagocytose it, and they also eliminate some dendritic spines. So as you can see here, this is a dendrite with the spines, so the little bulbs on it, and in yellow microglia. And I just posted this picture because the movie doesn't work. So you can see that microglia makes contact, is scanning, sitting on the dendrites. And as last, some sort of evidence is that microglia are implicated in synaptic maturation, because there is a study which knocked down or knocked out CX3CR1, so the fractalkine receptor, and I know Sander knows more about this. So they knocked it out, and they saw that the reactions of the neurons when they did patch clamp, the reactions were less mature. So that's quite big evidence that microglia play a role in the developmental synapse physiology. So now the last part. So microglia are also associated with radial cells. What are radial cells? They are only present during development. Radial cells are actually the precursors for astrocytes and neurons. So first they will generate astrocytes, and later in development they will generate neurons. Radial glia, they are called radial because here they are depicted in red. They have their cell soma located in the ventricular zone, and they have one very long process spanning to the pile surface and one really short one to the ventricular side. So the association with radial glia, so between microglia and radial glia, suggests that there is a functional interaction. And also because microglia colonize the brain before astroglial differentiation occurs, could suggest that microglia regulate the astrocytic differentiation. And there are some factors which have been found to regulate this in vitro, such as NO. This is actually based on a lot of in vitro evidence. But the other way around, you know that microglia, they ramify when they age. So it is also possible that the astrocytes control the microglial proliferation and ramification, because there are some factors like fibronectin and laminin, which are produced by the astrocytes, but also other factors. And a lot of these factors are just unknown, but they have shown to induce ramification or induce more the emiboid phenotype of microglia. For this part, there is only a lot of in vitro results, so nothing really in vivo. So the concluding remarks, as you all know by now, microglia are brain macrophages. So in adult, they have an important immunological function. And there is still ongoing debate about are microglia now beneficial or harmful to neurons. What are microglia now exactly, or is it microglia we see, or are they just infiltrating macrophages? And then during development, so in the beginning microglia derive from the Golgi sac, but it could be in later stages that they derive from blood circulating monocytes. And functions I discussed a lot, so blood vessel angiogenesis, tip cell fusion, the molecular mechanisms are now emerging, but for neurons and synaptic pruning and maturation, there are some suggestions, but the molecular mechanisms are still under investigation, and they associate closely with radial glia, but that field is quite open. So far the lecture. If you have any questions, I'm happy to answer them. Thanks for listening.
Ja, ik moet half op de auto achter.